thank you so much for allowing me to present today tell you a little bit about what we're doing here at Vascular Perfusion Solutions. The topic would be how to incorporate translational science and how at least we have incorporated the field of translational science into our small startup medical device company on our technology, which is called the VPS Encore device. Quick disclaimer, I'll be presenting some forward-looking statements. Just a disclaimer there. Okay. Oh, here. Can you guys see that?
Yeah.
Okay. So the area of research and medical technology that we're trying to get through has to do a lot with this statement that was given at the Organ Banking Summit and Harvard Medical School back in 2017. And it basically said that the ability to replace organs and tissues on demand could save or improve millions of lives each year globally and create public health benefits on par with curing cancer. So, that is a pretty big statement. It's a big challenge. And in order to make improvements in creating better outcomes and better solutions to save lives, especially on people that need organ transplants, it requires a big team of people. Kind of like, and I use this as an analogy. I have a mentor from Johnson & Johnson, and he always uses analogies of the Apollo program, where, you need the right team to take this to market and to be able to accomplish the goal. And just like the Apollo team was, conformed by many different types of people, very smart people, but it's also required a big team. And that's how we see things here in Vascular Perfusion Solutions. We have a big goal, which is be able to save lives by extending the life of organs. And we require a multidisciplinary team to be able to achieve that. And I focus a little bit on translational science and how that can, at least in my career and in the career of some of our employees here at VPS, has helped to be able to translate things. There's in the NIH, the National Institute of Health, has an institute called the National Center for Advancing the Translational Sciences, and it focuses on training individuals, physicians, and scientists on how to make the process more efficient. Most of the process is really focused on drug development, on how to identify drug candidates, and how to take them through a rigorous process of testing before it goes into the clinic, and how we can make that process more efficient. So that same process that is used for drug development, for translational science, can be used for medical devices. And that's something that we're trying to implement here. And I'll talk a little bit about the background of how the company came to be and how we're being able to implement the issue or the topic of translational science as a way of taking things to market. So what do we do here at Vascular Perfusion Solutions? One of the things is it's the shortage of organs and the organs that are rejected every year. As you can see, around 70 to 80% of hearts are rejected, around 30 to 40% of kidneys are rejected, 50 to 60 of livers are rejected, and 80 to 90% of lungs are rejected every year. As of November 2020, there were 114,000 people in the United States, patients waiting for heart transplants. This is the breakdown of each organ. As you can see, on hearts, around 3,700, kidneys, 94,000, livers around 13,000, and lungs around 1,356. So as you can see, this is a great need. There is a need for organs, not only in the United States, but everywhere around the world. So what is the current standard of care? The current standard of care for many years, ever since the first heart transplant occurred in South Africa, it's been taking the organs on ice. And that is really the standard of care around the world. You take the most precious organ out of your body, you put it on ice, and you only have a limited amount of time. With hearts, you have around 4 to 6 hours to be able to preserve the organ in the ice. And after that, we know that the longer the heart is remaining put on ice, the longer, the greater the failure of organ rejection. So for many years, this is the way that we collect organs and we keep them. So as you can see, it's limited. So what can we do to be able to improve that? We pull away from cold storage. If we have other technologies that we can extend the life of organs, well, we could have more organs available for patients. We can create better matches, because if you have longer periods of time to be able to better match those organs, you can basically have a better match between a donor and recipient, and that will lead to obviously less rejection. So what are we doing here? We have a medical device called the VPS Encore device. And the idea is that it preserves organs in a way that can extend the life of the organ. We believe that we can increase the organ availability of not only DBD hearts, which there are hearts that are usually now, most of the heart transplants that occur in the United States and around the world are from dead brain donors. So we're doing testing on those type of hearts, and we're also doing testing on donors after circulatory death, which are hearts that are basically stopped, and then you collect and you can preserve them. One of the achievements that we've done back in February of 2021, we were granted a breakthrough device designation. And in order to do this, we were able to gather all the data that has been collected by one of our founders. One of our founders was a professor at the University of Texas Health Science Center here in San Antonio. We took all the evidence from that. We've been also collecting some studies that we did in the last 2 years. We submitted all that to FDA, and our small little team of the startup was able to get a breakthrough device designation for our heart preservation machine. So we have also attained independent validation. So the system that we have is a hypothermic machine, perfusion machine. What it does, it circulates crystalloid solution at cold temperatures between 8 to 10 degrees Celsius. And by doing that, we're able to provide the essential nutrients that the heart needs to be able to remain viable. So some areas of collaboration that we've done is working with the military. When we were at the university before the company was created, we had a DoD grant where we tested our device on limb preservation, skeletal muscle. Now that we have moved on full time with the company, the inventor of the device as the chief scientific officer and myself as the director of translational science, we created all the areas of research and collaboration. One of those areas is with DARPA. We're testing a compound that allows for the metabolism to be reduced and to be able to extend the life of the organ for hopefully longer than 48 hours. And right now we have a grant through DARPA testing on this. So that's one area where of research. Our main area is the cardiac transplantation device, transport device. But another area of our research and development is to be able to expand on other areas of organ preservation. The parts of the intellectual property. So the first invention came out of the University of Texas Health Science Center, which is owned by the University of Texas system. The inventor of the technology has over 40 years of research and development on organ preservation. He is the former founder of Paragonix Technology, which is a company out of Boston. They do static cold preservation of hearts. Is the only 510K clear device for organ preservation for heart preservation, but that is a static device. It's not actively perfusing the heart, and it can only go for up to 4.5 hours. What we're trying to do, which the inventor basically invented a new device, and that patent was licensed exclusively by Vascular Perfusion Solutions, which is the first invention that we had, the first prototype. And with that, we started working on the device, and now we have actually our own patents on improvements of the technology and a whole different way of be able to extend the life of the organ even longer and more efficient than the original patent. So we still have the exclusive license from the University of Texas system, and we have our own patents now. And basically what we're doing as a startup, we're just trying to create new avenues for research and new inventions on how to be able to extend the life of organs beyond the standard of care. Hopefully what we're trying to do with the FDA right now is 8 hours, but we've done experiments in-house for up to 24 hours, and we're hoping to do even longer, 48 hours. And now with the new drug that we're working with Harvard and DARPA, we're hoping to do longer than that. So how we compare to what is out there? So we are not the only company trying to do this. There are several companies in this sector. One of our biggest companies, it's the company is called Transmedics. It's a pretty large device. What they do is they perfuse the organ with blood, and we don't perfuse with blood. We use a crystalloid, and they also perfuse the organ at physiological temperature. We don't do that. They also keep the organ um, basically pumping blood, which is different from us. So when we put our organ into our device, a heart into our device, we lower the metabolism by lower the temperature, and we just supply enough oxygen to keep the cells alive. So in case of a device, if our device fails during the preservation period, it basically remains in static cold storage, kind of reversing back to the standard of care. There are studies that we're doing to prove this to the FDA. And how we compare that with the competition is that if the heart ever fails during transportation, then the heart is lost because it's at a physiological temperature. They have gone through FDA. They had an advisory committee meeting a couple months ago. They're waiting to hear back from the FDA. Most of the advisory committee voted in favor, and the reason, and we believe in the transplant community, is that there's nothing out there really to preserve these hearts. And surgeons are desperate to be able to, extend the life of hearts and to be able to transport these hearts on a longer distance. So we believe that they're probably going to get acceptance and approval from the FDA because there really is nothing out there. And how do we see that as a company? We actually focus on hypothermic preservation of standard criteria hearts. Transmedics is going after extended criteria hearts. So it's a complete different, even though we're both in the same area, there are two different designations or different labels of use that we're trying to achieve. And so far, the results that we've done in lab show very promising. And I'll share some data on that about that from our experiments. So how do we think that what do we provide to the market? So we're hoping that with our machine, with our heart preservation machine, we can increase hospital profit margin. It is a $5 billion revenue opportunity. Hospital top line transplant revenue. Transplant centers around the United States are always trying to find better ways to do more transplants and have better results without rejection. So where does this is part of the history of where we come from. The inventor of the technology is Professor Leonid Bunnigan. He was a professor of anesthesiology at the University of Texas Health Science Center. Before that, he was at the University of Pittsburgh as a research scientist. He joined UT Health and started working on organ preservation. These are some of the articles that he's published before and how we were able to take this data and improve on it to be able to achieve the breakthrough device designation. He's done work on 12-hour preservation of hearts and in dogs and in different areas of also kidney preservation. These are some of the key heart preservation studies that we use for our breakthrough device designation. Well, the first study was done in 1996 with 10 dogs, and he compared that to cold static storage. So nice, and versus the device, they did an actual transplantation, and the hearts were able to perform normally for a few hours. They didn't follow this longer period than that. There was another study published in 1998, also on dogs, compared to cold static storage, and looking at different metabolic markers and oxygen consumption in the heart. There was another study in 2003, also on dogs, and what he did, he took hearts straight out of the dogs, put them on a Langendorf machine, which is a machine to be able to evaluate cardiac function, and compare that with the precursor of our device, and the precursor of the device had better results. We did a study back in 2019, taking DCD hearts and preserving them for 24 hours. We're about to publish this paper. And the new data that we've been working on for the FDA clearance is on DVD hearts. So this is ongoing research. We collect hearts from pigs. We put some on cold static storage for 4 hours, which is the standard of care. And our research arm is 8 hours on our cardiac preservation machine. These are some preliminary results that we have. These are hearts that are put before perfusion. This is after 8 hours and a half of perfusion. There's really no difference in the aspect or how the heart looks. One of the biggest things that we look for and what other scientists and transplant surgeons want to see is that there's no signs of edema. Sometimes when you perfuse on the heart can gain a lot of weight and a lot of water weight. So far, this is 7 hearts we've run, still preliminary data, but there's no sign of edema. The solution that we're using is a crystalloid, and the perfusions that we use are macroperfusions, close to less than 20 millimeters of mercury. And the results that we had so far are pretty convincing, and there's no sign of edema, which is a really, really good sign. One of the abilities or the things that our device can do is we're able to put our heart on our device on their C arm to be able to look at how the heart gets perfused. So you can see here a video of a heart inside a machine. We put some dye, and you can see how we perfuse the heart through the coronary arteries. This is a way to evaluate to ensure that the aortic valve is completely closed, and then the heart is getting proper perfusion. No other device can do that in the market. Most of the other devices are very complex, are pretty big, and our device, what we're trying to do is really keep it simple, small, and portable. This is some data showing lactate consumption. This is just like a metabolic marker that we look at that tells us that the heart is going from anaerobic to aerobic. So we're seeing that there's a conversion of production to consumption. And that's a really good sign for us to tell us that the heart is actually metabolically active during preservation, even at hypothermia. We can see oxygen consumption as well. We're doing some gene expression marker. We look at IL-6 expression, which is a major inflammatory marker. This is cholestatic storage. It's a high expression of this marker versus our device, which are, all of this is preliminary data, and it's good signs that we're on the right path. And then the way we're testing cardiac function is by putting the hearts on Langendorf, a machine, like I explained, that rewarms the heart at physiological temperature. And the heart on the machine can be able to be evaluated for cardiac function, cardiac stroke volume. This is just a video of a heart that was preserved on our device for 8 hours. This is ex vivo. We rewarm the heart, and you can see how it's beating. And this is just a graph showing contractility of a heart versus a fresh heart, and we're still collecting data on that. This is what we'll be presenting to the FDA. And who is part of our team? We have our CEO, is a Harvard alumni from the business school, and he was the founder of Paragonix, the first FDA-approved cardiac transport device. Leon Bunigan, our professor, Professor Bunigan, the CSO with 40 years of experience in organ preservation technology. Mark Mueller is our president. He is an engineer and a patent attorney for several companies, for Boston Scientific, HP, Intel, and eBay. Paul Perry is our CFO. Frank Guerra is our CEO of marketing. And then we have a small but growing team of scientists and engineers. Israel is a director of engineers, a fantastic engineer with over 20 years of experience working for Lysol, Allergan, J&J, and other companies. Myself, as another translational scientist with 15 years of experience, mostly focusing right now on our assays, of our experimental assays, immunological assays, and gene expression assays. And then we have our clinical director, who's a nurse. And we're still recruiting, we're growing, we're a small startup. And like I said, I think in the first talk, they're talking about having a good scientific advisory board. So we're creating that. So this is some of our scientific advisory board. And part of my job as a translational science director is creating partnerships with all the transplant surgeons. It is very important for us, as we are a small startup and are in development, that our device, it's in accordance to what the surgeons want and they want to see. We believe that the input from surgeons right on from the beginning is extremely important, because we don't want to have a device that surgeons won't be able to use. So the feedback that we get from them is fantastic. We have a growing collaboration with several transplant surgeons from UT Southwestern, from the Mayo Clinic, from other universities in the United States. We have some surgeons in Spain. We have some surgeons in Italy, and we keep growing. So that's part of also my job as a translational scientist, and be able to take the results that we see here in the lab, present that to to the transplant surgeons, so they'd be able to guide us and provide feedback on a device that can be useful and potentially save a lot of lives, like we're planning to do. These are some of our collaborators. Also, another line of research, like I mentioned, is we have some grants through Department of Defense, DARPA, the Harvard, the Wyss Institute at Harvard, local universities. We work with local OPOs where we're about to get human organs for testing, and all that data will be used for the FDA. The current path that we have right now is we're in a Series A fundraising. We're trying to raise money to finish our preclinical animal models and then our full clinical trial model. We are going to be a PMA path through the FDA, and we're working on that. And with that, I mean, it's a short talk, but if you have any questions, please let me know.
Well, thank you so much, Dr. Rafael, for your presentation and giving us a good overview of your company and showing us your team. And like you mentioned, the growth that you guys have had the past few years has been quite remarkable. And we'll give everyone a few minutes, see if anyone has any questions, comments, or for you. It doesn't look like we received any questions from anyone so far, but I see a lot of thumbs up coming, which is great. So, keep the thumbs up coming. Dr. Rafael will also be part of our panel later this afternoon at 4:35 p.m. EST, where they discuss AI and breakthrough technology. And definitely their organ-based technology is quite breakthrough. So yeah, I guess what we'll do is, since we're a little early, I guess we can all go to the next speaker room to set up. So we'll all head over to the next talk, which is Introduction of EU MDR.
Perfect. Thanks, Sam.